Hi, and welcome to this run-through for the 2018 AQA A-Level Biology Paper 2. And the reason I'm picking this paper to run through now is because this paper is probably going to be more similar to the paper that's going to come up in 2023. It's definitely going to be more similar than the one that came up in 2022, because if it came in 2022, it's less likely to come up in 2023, because they don't do the same questions like exactly the same 2 years running. So I haven't looked at this paper yet. I'm kind of going to do it with you live. I'm going to talk through my thought processes and then mark it. So this is a kind of a good thing for you to be doing in the final run-up to your exams. It kind of helps you understand the thought process that you need to go through when you're doing a paper and sort of to help you out. Just a little side note, it is 5 a.m. in the morning when I'm recording this, so apologies if I look a bit sleepy, but let's get going. Okay, question 1. Heat stress is a condition that often occurs in plants exposed to high temperatures for a prolonged period of time. Heat stress is a major factor in limiting the rate of photosynthesis. Okay, so this is the sort of new information that you're given. So you just need to kind of bank that, process that, and add that to your existing understanding of photosynthesis. Heat stress decreases the light-dependent reaction of photosynthesis. Okay, so you should know about this light-dependent reaction. Explain why this leads to a decrease in the light-independent reaction. So it's basically just asking you what's the link between the light-dependent and the light-independent. So if we decrease the light-dependent reaction, so we're going to have less ATP produced, and we're going to have less reduced NADP, which means that we can therefore do less reduction of triose phosphate, so less production of triose phosphate, less of the Calvin cycle, fewer turns of the Calvin cycle, and therefore fewer molecules of glucose produced. Okay, let's check the mark scheme and see if that's what gets us the marks. Okay, yeah, that's what we need. So ATP for the 1 mark and reduced NADP for the 2nd mark. Great. On we go. Okay, 1.2. Another effect of heat stress is a decrease in the activity of the enzyme rubisco. A decrease in the activity of an enzyme means the rate of the reaction it catalyzes becomes slower. A decrease in the activity of the enzyme rubisco would limit the rate of photosynthesis. Explain why. Okay, so the kind of image that you need in your head, you need to kind of have the Calvin cycle image in your head. So what this means, if the enzyme rubisco isn't working as well, is that we have fewer molecules of CO2 per second being fixed and attached to ribulose bisphosphate. So ribulose bisphosphate is this 5-carbon intermediate. We add the CO2 and we temporarily produce this 6-carbon intermediate, but then it breaks down into these 2 3-carbon molecules called glycerate phosphate, or GP. So fewer molecules of CO2 fixed to ribulose bisphosphate, and therefore making fewer molecules of GP. Let's check the mark scheme. Yeah, there it is. Let's zoom in here. Less carbon dioxide and less GP. Okay, 1.3. Where precisely is rubisco found in a cell? Well, this is found in the stroma of the chloroplast. That's where the light-independent reaction takes place. So it's just the stroma of the chloroplast. Double check mark scheme. It is early. Check I'm getting that right. Stroma, yeah, in the chloroplast. Now let's look and see what it rejects. This is kind of interesting. Reject stoma. Okay, so you've got to actually spell this right. A stoma is a bag that you might have to use if you have lower bowel problems. And it says reject stoma of cytoplasm or chlorophyll. Yeah, because these are all not quite correct. Reject mitochondrion. Ignore references to Calvin cycle or light-dependent reaction because that's not telling you where, it's just telling you what it's involved in. Okay, let's keep going. All right, here we get some experimental data. So whenever we get some experimental data like this, you read the read the spiel before. You're going to try and figure out what the graph shows you before you even get to the question. So scientists investigated the effect of temperature on the activity of 2 enzymes isolated from the leaf cells of cotton plants. Okay, so we've got rubisco and rubisco activase. That's interesting, an enzyme that activates rubisco. Figure 1 and figure 2. So So Rubisco activity seems to just keep, strange. Rubisco activity seems to just keep going up, 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 up with temperature. Doesn't seem to go down at all, even up to 50 degrees. Interesting. Whereas Rubisco activase seems to have an optimum temperature somewhere around 30 degrees. It says, the scientists concluded that heat stress reduces the activity of Rubisco in plant leaves by affecting Rubisco activase. Use all the information to evaluate their conclusion. Well, it seems right. Okay, so, all right, I'm just thinking in my head here. Okay, so, all right, evaluate. So normally evaluate, you want to have some things for the conclusion, some things against their conclusion. That's normally what they're looking for. So we're going to want to say some plus and minus things. Now, just glancing at it, I'm seeing more plus than minus, but potentially there's a little bit of minus. So first of all, things for their conclusion. Rubisco activity increases with temperature and does not appear to ever decrease. So Rubisco activity increases with temperature, reaching a maximum activity at 50 degrees C of 9. So I think we need to talk about that. It keeps going up. It does not seem... It would. You could also say, I think, that the optimum temperature for Rubisco, it must have an optimum, appears to be above 50 degrees. Okay, so potentially, if we kept going this way, it would sort of peak at some point, but it's above 50 degrees. So this is kind of interesting. We could also say, whereas the optimum temperature for Rubisco activase appears to be at 30 degrees C, so at temperatures above 30 degrees C, the activity of Rubisco activase is decreasing, and we could maybe talk about this is probably due to the fact that Rubisco activase becomes denatured. We might as well throw in the words tertiary structure, so the tertiary structure of the enzyme changes, the active site shape changes, and therefore substrate molecules may no longer fit in the Rubisco activase enzyme active site. So that's, I think, what we maybe can talk about for the sort of positive stuff. In terms of, I'll just put that down here. So, so it seems like, optimum temp Rubisco. I'm just going to write X is above 50 degrees C. It would appear to be. And then we can talk about the optimum temp or I just wrote RA, Rubisco activase. Now, what about the negative things? The negative things, you got to look, but you can start to spot patterns. They always do this. So the conclusion that you get written here, down here, which you have to evaluate, and often in exam questions, is too broad. Okay, that's a general point in these types of questions, because it says heat stress reduces the activity of Rubisco in plant leaves. Okay, in plant leaves, not cotton plant leaves, but all plant leaves. Now, is that fair? No, it's not. Okay, we haven't looked at all plant leaves. We're only looking at cotton plants. So that's too broad a conclusion. So we could say that the scientists concluded this happens in all plant leaves. However, the study was only carried out in cotton plants, so we can't generalize. We'd have to look at other plants to check. And I guess a final thing, I'm struggling here now because I've got one minus, but I need another minus because it's 4 marks and I want to say 2 positives and 2 negatives. The other thing that I might go for is that I might sort of throw this out there, there might be other factors involved. So we don't know. Heat stress may also affect other enzymes in photosynthesis. We're only looking at these 2 enzymes, but there's hundreds of enzymes involved in photosynthesis. So potentially, temperature affects other enzymes that aren't studied, and that's the main way that it reduces photosynthesis in plants. Okay, that's a tricky one. Let's have a look at the mark scheme and see if I'm on the right lines. Okay, here's the mark scheme. So, did I get this right? Okay, so first of all, Rubisco activity increases with temperature, or the Rubisco optimum temperature is above 50. Okay, cool. So that's the first thing we said. We said this, Rubisco activase activity decreases at high temperatures. Okay, cool. Optimum is somewhere around 30. I said 30. Yeah, look at all these things. Okay, so the negative things. It's only for cotton plants, that's what I said. Results graphs suggest activase. Why is that? I don't quite understand that. Results graphs suggest activase cannot or does not affect activity of Rubisco. Okay, so what they're saying is because the Rubisco graph keeps sloping up, it appears that its activity isn't affected, and it activase isn't affecting it at all because it keeps going up. I guess I was kind of thinking that these enzymes are somehow studied in isolation and not together, but I understand. This is what we said. For ice, there might be other enzymes involved. Is it what we said? I don't know. I might have only got 3 marks for this, 3 out of 4 for this. This is a tough question. So this is always a good one to remember. I forgot about this. Whenever they're saying, whenever they're making a conclusion, you can generally always put in, they haven't done any statistics, so we don't know if their conclusions are significant, or we don't know if the results are significant. That's a good thing to remember, which I just forgot. Right, let's keep going. Okay, question 2. There are different types of gene mutation. Put a tick in the box next to the statement which describes incorrectly the effect of the mutation in an exon of a gene. Okay, so we're looking for an incorrect statement about the exon. So remember, the exon is the actual protein coding part of the gene. Okay, so first of all, a substitution may not result in a change to the encoded amino acid. Okay, well that is true. I'm just going to write here true because that is referring to the fact that the code is degenerate. An inversion will result in the change in the number of DNA bases. This one is false. Now we'd still keep going and check these two, but it looks like this one is false. The reason, inversion just basically means a flipping. So if you take a number of bases and you flip it, look, I can use my daughter's colourful paint pots here. Imagine these are DNA bases. I flip it, I've got the same number of bases, they're just in a different order. So that one's false. A deletion will result in a frame shift. Yes, it will. Yes, it will. It will result in a frame shift unless it's a deletion of 3 bases and it kind of just takes one out. It doesn't really result in a frame shift, but this is probably true. Most of the time it's true. An addition will result in a frame shift. That's also true. So the answer is this one. Bink. 2.1. Double check. Mark scheme. Am I right? Hope so. Yep. Box 2. Cool. Ooh, nice descriptive one here. Describe how alterations to tumor suppressor genes can lead to the development of tumors. Right, so a tumor suppressor gene is a gene that is involved in regulating the cell cycle. So if we have an alteration in a tumor suppressor gene, it might mean that the cell cycle is unregulated, that mitosis happens more frequently, or the phrase we want to use is uncontrolled cell division. This might lead to cancer, especially if we start replicating cells that have mutations, because sometimes tumor suppressor genes are involved in basically checking for DNA damage and stopping mutated cells from dividing. And they may even send mutated cells into apoptosis, which is like controlled cell death. Okay, so that was 3 marks. I think I might have only got 2 there. So what do they want? So I said this, mutation of tumor suppressor genes, and then results in rapid or uncontrollable cell division. So what did I miss? This here, okay? So part 3, tumor suppressor genes are not transcribed or expressed. Yeah, I think this is, okay, I was going for this, and I should have said this, okay? So if there was a mutation in a tumor suppressor gene, then the primary structure, or even, actually look, there's lots of things we could have said here, or the tertiary structure would not be the same, and the protein may no longer work. Yeah. So you could have talked about methylation, which is a sort of epigenetic control. And I'm kind of surprised that this is in, oh no, this is paper 2, sorry. Yeah, you could have talked about methylation of the gene. Although my brain wouldn't have gone there because it said alterations to the genes. That for me implies mutation, but you could have talked about methylation of the gene, which would have reduced its expression through epigenetic means. Right, let's keep going. Ooh, maths. 2.3. A type of malignant tumour cell divides every 8 hours. Starting with one of these cells, how many tumour cells will be present after 4 weeks? Okay, this is fairly easy maths, but you need to be comfortable with standard form. Okay, I'm going to take you through it just to make it really clear, in case you're not sure. So if it can divide every 8 hours, how many times can it do in a day? Well, a day is, of course, 24 hours. And if you divide that by 8, you get 3. Okay, so the cell can divide 3 times in a day. And then we need to think about 4 weeks, or how many days is 4 weeks? Well, it's 4 times 7 days, so it's 28 days. All right, so we've got 28 days times 3 is how many times it can divide. So 28 times 3 equals 84. Okay, so if we had one cell, and then it divides after 8 hours, we'd have 2 cells, and then we'd divide, and then we'd have 4, and then we'd divide, and we'd have 8. So what is this? So at time 0, we have 1, and then this is 2 to the power 1, this is 2 to the power 2, this is 2 to the power 3, and so on. So 1, 2. So we're going to end up, at the end of the whole process, with 2 to the power 84 cells. Now we need to put that into our calculator and try and work it out, because we want it in standard form, not as a, I forget what this is called in maths. Is this a surd? I think it might be. We don't want to write it like that. We want to write it as 1 point something something times 10 to the something something. So, or whatever it is, 2 x to the y to the 84. That's going to be oh my gosh, that's a big number. Okay, so what we got is 1.93. I would probably put it to 2 decimal places. I wouldn't keep going. So I've got 1.934, so I'm going to round to 1.93, and that's times 10 to the 25. That is an enormous number of cells. Let's check it. Okay, yeah, 1.93 times 10 to the 25. If we had 84, 1 mark, or 28 times 3, 1 mark, we could have got incorrect answer, but shows 672 divided by 8. Okay, that's just the number of, what's 672? I don't even know how you would have got that, but this is the correct answer. Okay, on to question 3. Okay, so this is about respiration. Yeast cells can respire aerobically or anaerobically. Student used the apparatus shown in figure 3 to measure the rate of respiration in yeast. She positioned the flask in a water bath so that the yeast culture reached a constant temperature. Okay, so temperature is a control variable. So then she left the apparatus for 1 hour before starting her investigation. So this is like equilibration time. This is to make sure that kind of everything has reached the temperature of the water bath. Okay, so what have we got here? We've got coloured liquid, capillary tubing. Okay, so there's like a beaker within the conical flask. So that's the yeast culture in glucose. I presume that's like a glucose solution. And then we've got some alkaline pyrogalal to absorb oxygen. Okay, so to absorb oxygen, therefore this is going to be anaerobic respiration, because all the oxygen here is going to be like taken in. Okay. And also potentially as well, now that I've read this, it might take an hour for this pyrogalal to kind of absorb all the oxygen out of the air that is in the conical flask, so to make sure it's fully anaerobic. Okay, so
This is one reason why it was important the student left the apparatus for 1 hour after the yeast culture reached the constant temperature. So I presume this is that for the absorbing oxygen. So yeah, even after it reaches a constant temperature, they're still leaving it for another hour, and that is so that it can absorb all the oxygen, is what I think. Let's see if I'm right. Yeah, so the oxygen is absorbed or respired or used. Yeah. All right, 3.2. During her investigation, the coloured liquid moved to the right. Explain why it moved to the right. Okay, this is interesting. So this is kind of different to what you often see. So you often see this setup with something to absorb carbon dioxide, in which case the coloured liquid will move to the left as oxygen is used. But in this setup, the coloured liquid is going to move this way as CO2 is produced. So remember, all the oxygen is already gone from here. So yeast, when it respires anaerobically, produces CO2, and that's going to push this to the right. So we want to have probably the word equation, which is glucose goes to ethanol plus CO2, or carbon dioxide if I'm writing it in full. So we probably want to reference that word equation, say that it's respiring anaerobically. And then we probably also want to talk about pressure, I would imagine. So if we are adding molecules of CO2 into this beaker as they are released by the yeast, then the pressure in here is going to be slightly higher than the pressure outside, which is going to push the coloured liquid across to the right. Okay, so I think that is probably going to be sufficient for 2 marks. Let's see. Okay, yeah, so anaerobic respiration. We didn't need the word equation, but I think it's always good just to put it down just in case. And yeah, the pressure, you needed to talk about the pressure or the volume of gas increasing. Yeah, okay. Moving on. Okay, 3.3. The student found that the coloured liquid moved 1.5 centimetres in 24 hours. The diameter, this is important, the diameter of the lumen of the whole of the capillary tubing was 1 millimeters. The volume of the capillary tubing is given by pi r squared L, where pi is, they give you pi as 3.14, so that means they want you to use 3.14, not the value in your calculator, like 3.1419, whatever it is. So just use that. It says calculate the volume of gas produced in centimeters cubed per hour. Okay, and look at this as well. They're being tricky here because you've got multiple units. You've got centimeters cubed per hour. Over here we've got centimeters. Over here we've got millimeters, and here we've got 24 hours. Okay, so these are all the numbers that I use. Okay, so right, 1.5 centimeters in 24 hours. So first of all, let's work out what it did in a day, in 24 hours. Okay, well, 1.5 cm equals the length, but we want to do pi r squared times the length. Now, what is r? R is equal to 0.5 millimeters. Okay, did you spot that? Because this is the diameter of the tubing is 1 millimeter, so the R is 0.5 millimeters. So the volume of the capillary tubing that has kind of been pushed out is in centimeters. It's 1.5 centimeters times 0.05 centimeters. Okay, because I'm converting my millimeters into centimeters. This is, dividing by 10. Squared, because that's the radius, squared times pi. Okay, that will give me an answer, and then I'm going to need to divide it by 24 hours. So let's do that. So 1.5, right, let's do the squared first. 0.05 times 0.05 equals, okay, times 1.5 equals times 3.14 equals, it's not very much. That's in 24 hours, so I need to divide that by 24. divided by 24 equals, it's a very small number. It's really small, 0.00049. Now, it doesn't say you have to give it in standard form, but I think you probably should. So 4.91234 times 10 to the minus 4. Is that right? It feels quite small, but let's give it a go. 4.9 times 10 to the minus 4. Yes, it is. 4.9 times 10 to the minus 4. If you have divided by 24, even if you got an incorrect answer and you remember to divide by 24, that gets a mark. That's nice. I don't know what this bit is, but if you got any number in this range, then you get a mark. If you have the number 49, you get a mark. I'm not quite sure why that is. That's if you're doing working and you. I don't quite know why that is, but you might have got a mark for showing some intermediate steps of working, but not getting to the ultimate answer. It does say up here, it doesn't have to be in standard form. It says accept any equivalent mathematical representation of this answer. So you don't have to write it in standard form because it doesn't explicitly say. It could do, though. In a different question, it might say write in standard form. Okay, let's move on. Oh, these curves. I love these curves. Figure 4 shows a typical population growth curve for yeast under laboratory conditions. Okay, so by the way, just reminding you what these phases are called. This is the lag phase. This is when the yeast is kind of getting used to the environment, or it could be this could be the same curve for a bacterial population. So this may involve the yeast or the bacteria kind of switching on genes to effectively use the nutrients in its new environment, or just like getting accustomed to it. Then we have the log phase where the bacteria, or in this case the yeast, divide exponentially. Then we have the stationary phase. This is where essentially the replication of cells is matched by the death of the cells. So something is limiting as a factor limiting, and then the limiting factor becomes, I guess, greater. So then the population declines. Maybe the nutrients have run out, maybe oxygen's run out, something like that. Okay, a log scale is used to record the number of cells. I haven't seen this sort of question. Haven't got one of these types of question in OCR, which is what I'm used to. So I'm going to say it's because the yeast cells are replicating exponentially. I think I want to use the phrase exponentially. Or you could say, yeah, I think that's what I'm going to go for. Let's see if it's right. Exponentially. Okay. No, so, okay, this is what you need to say. Did not get this at all. There's a large range difference or increase in numbers. Okay, a large range or difference or increase in numbers. Something to remember the phraseology of that. Yeah, so for log scales, if you have an extremely large range of numbers, a linear scale becomes kind of impossible. So like, if this scale had numbers on it, like this was 1 and this was 2 and this was 3 and this was 4, etc., and this was 0, then we can go from 0, and this is 10 to the 4, so this is 10,000, and 10 to the 5 is 100,000, and so on. Okay, so we can have a big scale as opposed to just going from 0 to 4. If this is log, I'm going from 0 to 10,000, so a big range of numbers. Okay, many yeast cells die during the death phase. It suggests one reason why. Now this is interesting. You wouldn't say. My initial thoughts, if I hadn't remembered the rest of the question, might be because it had run out of oxygen. However, you have to remember the context of the whole question. You have to go back, scroll back up, and remind yourself that there isn't any oxygen to begin with, and this organism can respire anaerobically. So probably what is occurring is, as it's respiring anaerobically, a waste product builds up. That waste product is ethanol. So probably due to the production of ethanol. Now this is basically what happens when you produce like beer or wine, something like that that's fermented. Let's say wine. You've got grape juice. It's got a lot of sugar. The yeast grows exponentially, converting the sugar into ethanol. I guess you get a stationary phase where it's just, things are just about ticking along, and then the yeast cells start dying because the ethanol concentration has reached a toxic level, and maybe all the glucose has run out as well. So probably either of those would get the mark. Run out of glucose or build up of ethanol, I would imagine. Yeah, okay, so decrease or no glucose substrate. Feels a bit vague to me. Decrease or no glucose. I'm just going to get rid of that. I don't like that. Feels risky to say that. Or increase in ethanol or carbon dioxide or acidity. Except increase in toxins. That's what, yeah, ethanol is a toxin. Just a quick real world application of this. Wine. Why is wine like, what is it, 13% alcohol? That's basically the maximum concentration of alcohol that yeast can tolerate. So different strains, slightly different highest tolerances, but wine is somewhere like 11, 12, 13, maybe 14%, and then the yeast dies. Anything higher than that has to be distilled where the ethanol is concentrated. Oh, this looks like a fun formula. Ouch. Okay. Brace myself. Okay, the following equation can be used to make predictions of the growth in the population of yeast cells under ideal laboratory conditions. I'm going to underline that because I just feel like later on there'll be some sort of question that says, why did it not meet this, your estimated number? And it's probably going to be because the conditions weren't ideal, maybe. All right, what are the things here? Okay, xt, the population of after a certain time, so that's probably what I'm going to need to kind of work out. X0, the population at the start. E, base of natural logarithms. R, growth rate. T, time period in hours over which R applies. Okay. A population of 2,000 yeast cells was left for 10 hours. The value for the growth rate was 0.5. Okay, so this should be xt is equal to x naught. I'm just writing this out again, just so I don't go wrong. So x naught is 2,000. That's the starting population. Times e, which is 2.72, to the power of r times t. So the growth rate is 0.5 and to 10. So it's 2,000 times e to the 5. Okay, so I'm going to do that on my calculator. So remember, e is 2.72, so what I'm going to do is going to go put 2.72. You've probably got this on your arithmetic, your scientific calculator, but I'm going to do 2.72. I'm going to press this sort of x to the y button, and I'm going to do it to the power of 5. It gives me 148, 149, and then I'm going to times it by 2,000. So that gives me 297,765.59. Doesn't tell me to do it standard form or anything like that. So I guess I'll write it in full. I might write both just to be safe. So I'd write 297,766. Okay, is the rounded up because it's 0.59, and then maybe I put in brackets here. I don't know. This is kind of insurance. 2. Oh, hang on a minute. 3 times 10 to the 5 to 2 significant. So 3.0 times 10 to the 5. Actually, I should have probably written. Let's see if I get marks for either of those. 3.0 times 10 to the 5. Yeah, I get it. Okay, 297,766. What do they also accept? 2.98 times 10 to the 5. Or I guess. Yeah, I think those all would have been credited. Yeah, with appropriate rounding. So I think 3.0 times 10 to the 5 would also be correct. Okay. Let's keep going. Ooh, right. This is a lovely graph. Let's look at this question. Okay, 4. Scientists investigated the process of succession on sand dunes. They measured the percentage cover of different species of plants on sand dunes of different ages. Some of the results the scientists obtained are shown in figure 5. So sand dunes are often studied for succession because basically as you move away from the coast inland, you're moving away like in distance, but you're kind of also moving back in time, because as the further away you go from the sea, the longer the dunes have been there, because basically the sand accumulates and the kind of coastline moves out into the sea as more sand accumulates in these kind of duney places. Okay, I'm going to get to that question in a minute, but I first of all just want to look at this and really like try and understand it. So we've got mean percentage cover. So this is going to be kind of quadrat based. And then we're kind of looking at age of sand dune down here. Now what's going on? Okay, so I'm looking at beach grass is the first one. It's the first highest peak here. I'm thinking that this is some sort of pioneer species because it's in the early phase. And then we've got this shrub bunch grass, which kind of comes in second. That's like a transitional type thing. And then we're getting conifer trees and then hardwood trees are the latest thing to arrive. So describe how you would determine the mean percentage cover for beach grass on a sand dune. Okay, gosh, what am I expecting to be here for 3 marks? So notice it says mean percentage cover. So that means we're going to have to do repeats. So I think this is all about quadrats. So I think that what we're going to get a mark for saying, we'll go to the mark scheme in a second, but I'm thinking I'm going to lay out axes on a patch of sand dunes. I'm going to use random number generators to generate an x coordinate and a y coordinate. I'm going to place a quadrat where those 2 coordinates meet. I'm going to look in the quadrat and estimate the percentage cover of the beach grass. I may even use a quadrat which has subdivisions. So sometimes you get a quadrat with 25 small squares, and then each small square is worth 4% of the cover. So I could count the small squares to work out the percentage cover in that quadrat. I would repeat this process for multiple quadrats, maybe let's say 10 quadrats, and then I would calculate the average percentage cover. Okay, I think I probably said about 5 things there, so I should get marks for that. What's on the mark scheme? Okay, so a method for randomly determining the position, so a random number generator, large number of quadrats. Now, how many? I wouldn't have got this mark. Look, okay, if a specific number is given, it must be 20 or more. Okay, let's just try and bank that in our memories. If ever you have to talk about doing an investigation with quadrats, you could either just say large number of quadrats or more than 20. Let's just have that in our head. More than 20 quadrats, always in an ecology investigation for this spec. Number 3, divide total percentage by number of quadrats. Yeah, okay. So this is basically just telling you how to work out a mean. Yeah, okay. To work out a mean, you need to add them all up and divide by the number of quadrats. Is kind of what this mark point is for. So I think I would have only got one. I think I only would have got 1 out of 3 there, even though I said all that stuff. Okay, so when you are another exam technique point, I'm new to this spec, as you are, I presume. If it asks you specifically to work out a mean, you really need to articulate how to work out the mean. You can't really take it for granted. So you add up all the percentages and divide by the number of quadrats. Okay, so if it asks for a mean, explain how to work out a mean. I feel it feels kind of GCSE, but you need to do that. The scientists concluded that results shown in figure 5 were due to succession taking place. Okay, use figure 5 to explain why scientists reached this conclusion. Okay, so where do I think marks are going to be given. So, okay, so first of all, you want to talk about the beach grass. Okay, you want to talk about beach grass is a pioneer species. It maybe reference this peak here. It's most abundant at about 25 years. Let's take a step back, actually. So I'd say initially, as the dune is 0 years old, there does not appear to be any vegetation at all. But quite rapidly, a pioneer species comes in. That pioneer species is beach grass. Beach grass becomes the dominant species with a maximum percentage cover of 30% at age around 25 years. You could then say this pioneer species starts to change the character of the dune, maybe causes the dune to have more minerals in the soil, and that allows other species to come in. And then talk about the second species. Shrub bunchgrass is a transitional species that becomes very abundant and changes the character of the sand dune, changes the nutrient levels in the sand, so that then conifer trees can come in. As the conifer trees come in, they outcompete, this is key language, they outcompete the shrub bunchgrass, so that the percentage cover of shrub bunchgrass goes down, and the conifer species become dominant up until about 400 years. Then you could say after 400 years, the conifer species percentage cover begins to decrease as hardwood trees outcompete the conifers. Maybe you could then say ultimately, after a few thousand years, you might expect the hardwood trees to be the climax community. That is the final stage of succession. That's quite a lot to write, but it is worth 4 marks. So you write stuff, try and get all marks. Let's see where the marks are. Pioneer species, that's 1 mark. Okay, it changes the environment. For example, more humus or nutrients. Humus, by the way, not to be confused with humus. Humus is the biological material in the soil. Humus is a spread that you make with chickpeas. Okay, humus has 2 M's. Humus has 1 M. Okay, the pioneer species changes the habitat, so it's less hostile, more suitable. I think this is phraseology that I want to remember. It's more suitable for the named species. Yeah, and then there is that point as well for talking about the climax community. You could actually, they give you a mark for either saying conifers or hardwoods are the climax community. Okay. Let's keep going. We're doing good. Ooh, what's this? The scientists also investigated how the proportion of sunlight reaching the ground changed during succession. Okay, now the first thing I notice here is obviously the sunlight goes down, but also I notice the scale is different here. Look at this. This is 500 years. This is kind of, and up here, it only goes to 500 years. Okay, so the scale is much bigger. So this all of this kind of journey from 0 to 500 is going on here, and that's really where the change occurs. So when there's the pioneer species, all the light's reaching the ground because the pioneer species is a grass, and then as shrubs and other trees grow, less and less light reaches the ground. So that's basically what you want to say. The trees block the sunlight. There it is. Trees block or reduce the sunlight. 4.4. Using evidence from figure 6, that's this one up here, what can you conclude about the net primary productivity, NPP, in the sand dunes that are older than 1,000 years? I don't know. Well, there's no change in the amount of sunlight reaching the ground after 1,000 years. This graph is flat. So I presume there must therefore be no change in the primary productivity. And the explain is because there is no change in the sunlight reaching the ground, there must be no change in the amount of sunlight being captured by the trees, and therefore the NPP of the trees must be the same. I don't know about that. Okay, so I said this, NPP remains constant. I didn't say this. I didn't say photosynthesis remains constant. And respiration. Does it say GPP? Net primary productivity. Oh yeah, GPP is gross primary productivity. Yeah, I haven't revised this recently. So I would have got just one mark for NPP remains constant. What is the explanation? So gross primary productivity and respiration is constant. Oh yes, I remember this now. So these two terms are almost synonymous. Oops, let me get rid of that. So GPP is essentially equal to photosynthesis. Okay, GPP is your gross primary productivity. How much sugar is made, and it's by photosynthesis. So we can kind of use those terms interchangeably in this question. So GPP or photosynthesis is balanced by respiration. So therefore it's constant. Okay. Okay, on to question 5. So, furosemide and CBT are drugs used to remove excess fluid from the body. Scientists investigated the effect of these drugs on the volume of urine produced by human volunteers, and they used the following method. 3 groups, A, B, C, at random. All volunteers the same food for 3 days, I guess prior to the start of the test. After 3 days, they gave the volunteers in group A a tablet containing furosemide, B, CBT, and C, they had a placebo. They then found the mean volume of urine produced by each group in the 4 hours after taking the tablets. Okay? Group A, furosemide 1980, Group B, CBT 1201, and Group C 312. Okay, all right. So it seems fairly clear. Oh, this is plus or minus 2 standard deviations. Okay, didn't spot that. All the volunteers are given the same food 3 days. Okay. All right, so why were they given the same food? They were given the same food so that there was the same... They took in the same amount of mineral ions and salts, I suppose, because this would affect... potentially affect the water potential of their plasma, their blood plasma, and it might affect their kidney function. That's what I'm guessing. It doesn't actually say they were given the same drink, but I presume they were given the same drink and food. Let's see what the mark scheme says. Okay, because the foods would affect the water potential of the blood or body. I said blood plasma. And therefore it might affect the volume of urine produced. I think I would have only got 1 there. It might affect the volume of urine produced. (5.2) Using Table 1, what can you conclude about the effectiveness of furosemide and CBT in the removal of excess fluid from the body? Well, okay, 2 marks. It seems like they're both effective. So I would say that both furosemide and CBT are effective in removing excess fluid from the body. It seems like furosemide is the most effective at removing excess food from the body. And also, I think you could say it appears that the results are significant as the standard deviations do not overlap. However, we might need to do a statistical test to confirm this, is what I would say. Let's look at the mark scheme. Okay, so furosemide and CBT are effective. Furosemide is more effective than CBT. Yeah, difference as standard deviations do not overlap. And you do need to say more effective than placebo. They are more effective than the placebo, than the control group C. I think I would have got 2 because I would have got this one and this one. More comparative statements, more effective than. 5.2. Using Table 1, what can you conclude about the effectiveness of furosemide and CBT? I've done that one already, sorry. 5.3. Furosemide is sometimes used to treat high blood pressure. Suggest how furosemide would cause a decrease in blood pressure. Well, this is simply if you remove fluid from the blood plasma, there is a lower volume of blood plasma and therefore a lower blood pressure. So it lowers the volume of the blood. Lowers the volume of the blood. 5.4. Furosemide inhibits the absorption of sodium and chloride ions from the filtrate produced in the nephrons. Okay, so I just quickly dropped this picture in here just to explain. So if you inhibit this absorption of sodium and chloride ions, so you're kind of inhibiting the absorption of these ions and these ions from the lumen into the blood plasma, then that means there's going to be a lower concentration of sodium and chloride ions in the blood plasma in the nephron capillaries, and therefore there will be a higher water potential and then less water. There'll be less of a water potential gradient, so less water will be reabsorbed from the glomerular filtrate. That's what we need to say. So water potential of the less water. Did I say osmosis? Okay, so here's the mark scheme. Now what I said was, I mentioned this water potential gradient, so I think I would have got 0.1. I said the water potential of the water potential of the blood would be increased, so I think that's kind of the same thing as the water potential of the filtrate would be decreased because there's more sodium and more chlorine ions staying in the filtrate. I don't know if I used the word osmosis, which was very silly of me. I think I did, actually. Maybe I did. But remember, you always have to say osmosis. Even if your brain is thinking, oh yeah, it's osmosis, water's moving from here to here, you have to say the word by osmosis, always, by osmosis. And you could have talked about the collecting duct. I actually interpreted this as the PCT. So the proximal convoluted tubule or distal convoluted tubule or loop of Henle. You could have talked about all of those. It says ignore PCT. So if you write proximal convoluted tubule, you need to write it in full, not PCT. So there's the mark scheme, and let's keep going. The scientists also measured the mean rate of flow of blood plasma into the kidneys. Here are the results. So we've got 380, 342, and 295. Okay. The mean rate of flow of blood plasma is 60% of the mean rate of blood flow into the kidneys. So what that's saying is essentially that blood is 40% cells and 60% plasma, I think. How much greater is the flow of blood into the kidneys with furosemide than with group C? And this is blood plasma. Okay, so if we're trying to work out the rate of flow of blood, not blood plasma, what we need to do is divide it by 0.6, which will give us a bigger number. So divide that by 0.6, divide that by 0.6, and we're going to get a bigger number. What have I done with my calculator? Phone. Okay, so I do 380 divided by 0.6. 380 divided by 0.6 equals 633.3. And then 295 divided by 0.6 equals 491.6. And it says how much greater is the flow of blood into the kidneys with furosemide than with group C. So I do 633.3. 633.3 minus 491.6. I got 141.7. Gives your answers in centimeters cubed. Oh, okay. This is hard for 1 mark. This is hard because that's 141.7. But what have I worked out? This is centimeters cubed per minute. So I need, and it says over the 4 hours of the investigation. So I need to times this by 60 and I need to times it by 4. That would be very easy to not see. Is that 34,008? Right. Was that right? Let's see. Yes. Yes, it is. Somewhere between 33,840 to 34,680. That's a hard one for 1 mark. I find that's quite. I'm quite amazed that's a 1 marker. I would have thought it would have been 2. Okay, on to question 6. In genetic crosses, the observed phenotypic ratios obtained in the offspring are often not the same as the expected ratios. Okay, so first of all, fertilization is random. So you won't get exactly what you're predicting. This is kind of the average. If you were to do the cross multiple times, you get about this ratio, but you're going to get a little bit either side. So fertilization is random. Fertilization is due to chance. Now, this is a list principle thing. So you need to pick your 2 most secure answers. If you give 3 answers, then they're not going to mark all 3. They're only going to mark the first 2. And in fact, if you get the 3rd one wrong, it can actually take off a mark. So fertilization is random, and the other thing I'm thinking is maybe there might be crossing over, or there might be mutation. What would I go with? I guess I'd go there might be crossing over. That's what I'm going to go with. I'm not sure if that's going to get me my 2nd mark, but I give it a shot. Look at all these things. Could have had any of these. Okay, this is the one I said. Fertilization of gametes is random. Look at all these things you could have said. Small sample size, genes may be linked. So this is, you could have written autosomal linkage. Oh, I would have got a mark, thankfully. It does say except crossing over, so I would have got a mark there. But there are much more easy things I could have said, possibly. Epistasis, lethal genotypes. So if a genotype is lethal, like if a double recessive is lethal, then you do your cross, you're not going to have any double recessives. Like, where are those 1/4 double recessives? Well, they all died before they were born. So that could be the case. All right. Possibly worth memorizing those different answers you could have given. All right, so now we have a cross. Tomato plants. The genes for height and for the type of leaf are on the same homologous pair of chromosomes. So this is going to be about crossing over, or maybe or like autosomal linkage. The allele T for a tall plant is dominant to the allele small t for a dwarf plant. The allele M for normal leaves is dominant to the allele little m for mottled leaves. Okay. Cross between parent plants, heterozygous for both genes, and examine the offspring produced. The position of the 2 alleles for both genes was the same in each parent plant, as shown in figure 5.7. Okay, so we start with like both dominant alleles on the one chromosome and both recessive alleles on the other chromosome. Okay, so both parent plants are like this. Okay, so I'm looking at this, and basically this is about crossing over. So you can see that, you know, tall plants are normal leaves. Even before I get to the question, this is kind of like inheriting at least one of this version of the chromosome. Like if you inherit one of those chromosomes, then you're going to have tall plants and normal leaves. If you tall plants and mottled leaves, to get that you're going to have some crossing over. Dwarf plants and normal leaves, to get that you're going to have crossing over. Dwarf plants and mottled leaves, you're going to have 2 of these versions of the chromosome. What would the genotype of the offspring with dwarf plants and mottled leaves be? So that's this bottom one. It's going to be little t, little m, dwarf plants and mottled leaves. Okay, what's the next question? Okay, use the information provided to explain the results in table 3. Okay, so I kind of actually just did that, didn't I? So that's what you need to say. So what do you need to say? Well, I would say that the two alleles are on the same chromosome and therefore show autosomal linkage. And you would say it's more common for both dominant alleles to be inherited together or for both recessive alleles to be inherited together. So, and then you would say that the tall, to have a tall plant with normal leaves, it could either be kind of inheriting a big TM chromosome plus another big TM chromosome to give you TTMM, or you could get one big T, big M chromosome and one little t, little m chromosome, but that would still give you tall plants and normal leaves, and then you would sort of elaborate about the crossing over. So let's see the mark scheme points for that. Okay, so linkage produces few tall, mottled, or dwarf normal offspring because this is crossing over has to occur for this to happen. Alleles are linked, except produces few TM and so TM gametes. So these are called recombinants. You would probably call these recombinant gametes. So if crossing over has occurred, let's go back to this one, between at any point between these two gene loci, then you could get a chromosome that looks like this, with, let's say, a big T and a little m. And of course, if crossing over did occur like that, then the other chromosome that you would get would have a little t and a big M, because crossing over has exchanged versions of the alleles, and these would be called recombinant chromosomes. Recombinant gametes. Moving on to 6.4. Complete table 4 to show the expected ratio of phenotypes if the same cross had been carried out, but the genes for height of plant and for type of leaf were on different homologous pairs of chromosomes. Okay, so this is basically like no linkage. So this is two different alleles that are not linked, two different genes, sorry, that are not linked. So we would get the standard ratio, which would be 9 to 3 to 3 to 1. So 9 for the double dominant characteristic, like dominant and recessive characteristic, recessive and dominant characteristic, double recessive, both recessive characteristics. So expected ratio. So I think that's just, I guess 1 mark for each column. So 9 to 3 to 3 to 1, and then you just write tall, normal, etc. Okay, so tall plus normal, etc. It does say the ratio. I was just checking that it doesn't actually want the numbers. Okay, yeah, 9 to 3 to 3 to 1. That's correct. Moving on. Ooh, question 7. Okay. Dopamine is a neurotransmitter released in some synapses in the brain. The transmission of dopamine is similar to that of acetylcholine. Dopamine stimulates the production of nerve impulses in the postsynaptic neurons. Describe how. Okay, so basically this is just asking you how to explain how a nerve initiates an action potential like at a synapse. It says, do not include in your answer the events leading to the release of dopamine and the events following production of nerve impulses at the postsynaptic neuron. So you have to limit yourself to the actual synapse. Okay, so dopamine, maybe I'd start just a little bit earlier just in case. So I say dopamine is released from the presynaptic neuron. It diffuses across the synaptic cleft, binds to receptors on the postsynaptic membrane. These receptors are ion channels, and when the dopamine binds, it opens the ion channels, causes let's say sodium ions, probably is sodium ions, causes sodium ions to flood into the postsynaptic neuron. This depolarizes the postsynaptic neuron, it reaches threshold potential, and then an action potential is triggered. Okay, and that should be plenty for 3 marks. Let's see if that's what it is. Yeah, so diffuse it. Look at this. There's a lot of highlighted words here. So you've really got to use your language specifically or correctly. Diffuses. Okay, receptors on the postsynaptic membrane, sodium ions, and causes an action potential. Okay, great. All right. Seven point two. Dopamine has a role in numerous processes in the brain, including pain relief. The release of dopamine can be stimulated by chemicals called endorphins produced in the brain. Endorphins attach to opioid receptors on presynaptic neurons that release dopamine. Let me read that again. Dopamine is okay. The release of dopamine can be stimulated by chemicals called endorphins. So endorphins attach to opioid receptors. Okay, so the endorphins attach to opioid receptors on presynaptic neurons, and then that causes them to release dopamine. Morphine is a drug that has a similar structure to endorphins and can provide pain relief. So that's a weird one. You almost have to go just like rewrite this, but then talk about morphine. So morphine can attach to these same opioid receptors on the presynaptic neurons and trigger the release of dopamine. That then it provides pain relief. Is that it? It feels a bit like not enough. Yeah, that is it. So morphine attaches to the opioid receptors because it has the same shape, and therefore dopamine is released. Okay, yeah, that's pretty simple. 7.3. GABA is a neurotransmitter released in some inhibitory synapses in the brain. GABA causes negatively charged chloride ions to enter the postsynaptic neurons. Okay, so here's a picture of this going on. How does this inhibit postsynaptic neurons? So with the opening of chloride ion channels, chloride ions flow from the extracellular space into the postsynaptic neurons. This makes them become hyperpolarized, so the membrane potential becomes more negative, which means it's then harder to reach threshold potential, and it's harder for those postsynaptic neurons to initiate an action potential. And in fact, they kind of cancel out. Like if you can actually get, can't remember if this is on your spec, I think it is. If you have an excitatory postsynaptic potential, so like an acetylcholine neuron stimulating the postsynaptic neuron, and you also have a GABA neuron stimulating a postsynaptic neuron, they kind of cancel each other out. The GABA produces an IPSP, inhibitory postsynaptic potential, and the acetylcholine one produces an EPSP, and they can kind of cancel each other out. So let's check the mark scheme. So more negative or hyperpolarization, the words you need to say. So it's I said it would be harder for depolarization. I don't know if that is quite good enough. So yeah, more sodium ions would be required for depolarization. Okay, that's what I didn't maybe clearly say this enough. More sodium ions would be required to initiate or to reach threshold for depolarization. Moving on. Question 8. What is meant by a genome? So genome, this is just a definition. This is the total genetic code of an organism. I think that would get me the mark. Total genetic code of an organism. All the DNA in an organism. I think so. All the genes, alleles, genetic code in an organism. Yeah. Okay. Yeah, that's good. We would have got that. But here's the definition, the easiest one. The DNA in an I think you do need all, to be honest, for the definition. I wouldn't put all in brackets. I would say always say all for the genome, all the DNA in an organism. Okay. Chromatin immunoprecipitation is one method to determine where a transcription factor binds to DNA. Okay, here is the principle. All right. Add chemicals to bind the transcription factor to DNA. Okay. Break down DNA into small fragments and add an antibody specific to the transcription factor. Remove the precipitated DNA. Okay, fine. Okay, so basically, I'm going to try and draw this diagrammatically. So a big old long stretch of DNA. Transcription factor binds. This is my transcription factor, this blob here, TF. Then we break it down into small fragments. So I kind of like chop it into small fragments. These blue lines are like my little breaks. And then remove the precipitated DNA. You use that antibody to just pull out this bit. And add a chemical that makes the antibody. Sorry. Then you use an antibody to precipitate it. I'm going to put the antibody on there. And then add a chemical to remove this bit. And you've just got this bit, the DNA, and then you amplify it, and then you've got loads of copies of this little sequence like that, which kind of shows you the sequence that binds to the transcription factor. Okay, so that's how it works. All right, questions. What are the questions on this? Explain why the antibody binds to the transcription factor. So this is kind of almost like an immunity kind of question. So what do we need to say? Well, what part of the antibody binds? I would say that the variable region of the antibody is complementary to part of the transcription factor molecule. And I might also say this means that its tertiary structure is a match for the tertiary structure of the transcription factor molecule. I think that's it. Let's have a look. Yeah, okay, so the two things that I said, variable region, and then I was talking about the tertiary structure because I wasn't quite sure which way the question wanted me to go, but if I would have mentioned both, and actually either one is accepted, and then the other thing I just said was it's complementary. Remember that, like, don't necessarily talk about active sites. Don't make the mistake, of talking about active sites. Like active sites are only enzymes. Just because shapes match doesn't mean something's an active site. Complementary is a term that you can use for enzymes and for antibodies and for other things, not active sites. Okay. Whoops. What did I do? Okay, 8.3. Use figure 8 to explain what precipitated DNA consists of. Okay, so yeah, I had to kind of figure this out, and I kind of drew it to kind of understand it. That's a technique you could use. To be honest, I had never heard of chromatin immunoprecipitation before. Maybe I should have done, but I had to kind of try and work it out through like picturing it in my mind, and for me, just having the words there was not helpful for me because I kind of think more pictorially, so I had to kind of draw it to understand it. But now I think I do understand it. So the precipitated DNA, what does it consist of? So this is the DNA and the transcription factor and the antibody all attached to each other, because this is the precipitated DNA, and then you add the chemical that removes the antibody with the transcription factor. Okay, so I would say precipitated DNA consists of the antibody attached to the transcription factor attached to the DNA that it binds to. Is that correct? Yeah. DNA, transcription factor, and the antibody. All three of them together. All right. Now a little bit of text to read, so let's analyze. Soybeans are used in a number of processed foods. However, soybeans contain a protein known as P34 that causes an allergic response in some people. Scientists have created transgenic soybeans that produce single-stranded cDNA, which prevents transcription of the P34 gene. They use recombinant plasmids as vectors to transform soybean cells. After they had screened these cells for production of the P34 protein, they cultured the transformed cells to form soybean plants. How could it prevent transcription of the P34 gene? So basically, the single-stranded cDNA probably produces a short, so it's siRNA basically, so a short interfering RNA molecule that has a sequence that is complementary to the messenger RNA for the P34 gene. So the siRNA and the mRNA come together, forming double-stranded RNA that is then degraded, preventing the translation of the P34 protein. So therefore, yeah, it binds to the P34 messenger RNA. Yeah, well, you only needed to say one little bit, but that's how it would work. Okay, moving on. Describe the roles of two named enzymes used to insert DNA fragments into plasmids. Okay, so the two things that we're gonna want to mention are restriction enzymes and DNA ligase. Okay, restriction enzymes cut the DNA sequence at a specific site, and then maybe I might go on to say forming a sticky end. I might even draw a little picture of a sticky end like this. I mean, sometimes it's rare that pictures are credited, but I might just illustrate anyway. And then ligase forms phosphodiester bonds joining up the backbone. Okay, so that's the two things that I would say. Let's see if those right. So restriction enzyme to cut the plasmid, ligase joins the DNA. Yeah. .6. The soybean cells were screened for the presence of the P34 protein. This process involved the use of gel electrophoresis to separate proteins extracted from soybean cells. So just two features of the structure of different proteins that enable them to be separated by gel electrophoresis. So basically this is asking you, why does gel electrophoresis kind of work? Okay, so proteins can be different sizes, and proteins are generally charged. Is that right? Yeah, okay. I was pretty skeptical about that, so I wasn't really sure, but yeah. Oh, ignore size. Ignore size. Size is not a good enough word. I said different sizes. Different masses, okay? So proteins have a different mass or different number of amino acids, and they also have different charges because of their differing R groups. Could have said different tertiary structure. So that means that they can be separated out by the electrophoresis because they will, because of their different charges, they'll move at different rates towards the negative electrode. And because of their sizes, they'll move at different rates. Bigger proteins will take a longer time to kind of work their way through the electrophoresis gel compared to smaller proteins. I said size again. Heavier, more massive proteins with more amino acids will take longer to move through the gel. Okay, so top exam tip I need to remember next time I do one of these papers: don't mention size of protein. Talk about number of amino acids in the protein if you're talking about electrophoresis. Size is not credited. Okay, question 9. Lactose is the main sugar in milk and is hydrolyzed by the enzyme lactose. So lactose and lactase, don't get them muddled. Lactase is essential to newborn mammals. So all newborn mammals have it, as milk is their only source of food. Most mammals stop producing lactose when they start feeding on other food sources. Humans are an exception to this because some continue to produce lactase as adults. So like the gene never gets turned off, it kind of keeps being made. The ability to continue producing lactase is known as lactase persistence, LP, and is controlled by a dominant allele. A number of hypotheses based on different selection pressures have been put forward to explain lactose persistence in humans. One hypothesis for LP in humans suggests that selective pressure was related to some human populations farming cattle as a source of milk. Yeah, okay, so 4 marks. So farming cattle as a source of milk. This means kind of calories from milk available. Now we need to say, prior to the farming of cattle for milk, there must have been mutations. There must have been a mutation in the population that meant that some human individuals were able, or some human individuals kept producing lactase. When more calories from milk became available after the advent of farming, this mutation gave an advantage. Okay, because of it enabled them to get more calories, to be healthier, and they had a greater chance of survival. They then passed on their genes, leading to an increase in the allele frequency, which results in evolution. Furthermore, you could say that the presence of cattle being farmed for milk acted as a strong selective pressure. Yeah, if we use those terms, we should definitely get 4 marks. Let's see how we do. All right, mutation. Oh, named nutrient. I said calories, but okay, probably wouldn't have got that one. But any of those named nutrients, glucose, galactose, or protein, survive and reproduce, pass on their genes. Allele frequency. I would have got this one. This one. Okay, for number 3, pass on the allele is acceptable for reproduce. I was worried that I would have had to say reproduce, but pass on the allele. Just as a thing that I'm going to remember when I answer more, I would probably phrase it something like, they're more likely to survive, reproduce, and pass on their alleles. I'm just like really going to like try and make sure that I always say exactly what they want, because it is different examples, so quite picky with what they want you to say. Okay, there's the mark scheme. See how you did. (9.2). Use the information provided to explain why the number of people showing LP would rapidly increase once selection for this condition had been established. Well, I would say it's a strong selective pressure, which conferred a greater survival advantage. So the allele frequency would change quickly, and the other thing to say is, because the allele is dominant, individuals only need to inherit one copy of this dominant allele to show lactose persistence. That's probably what I would say. So try and mention those 2 things, kind of strong selective pressure and dominance, I think is probably where we need to go with this. No. Okay, so we need. Well, I probably would have explained that. So the fact that it's dominant, and then kind of basically this is just unpacking what dominant means. Dominant means it's expressed when there's only one copy of the allele present. So yeah, I probably would have got that. I think that's where I was going. Okay, on we go. 9.3. Lactase persistence is caused by mutation in DNA. This mutation does not occur in the gene coding for lactase. Okay, so the enzyme lactase is present in all humans, in all human babies. So it's not a mutation in the actual enzyme itself, it's a mutation in the promoter region that controls when the enzyme is made and when it's not made. So I suppose prior to this mutation, the promoter was under some control so that the promoter would only switch the gene on early on in your life, first few months of life, and then it would turn off once you start eating kind of like food. But the mutation would be in the promoter such that the promoter is now kind of always on and isn't switched off. Okay, does that make sense? So it's a change in the promoter, not the gene. So mutation in the promoter, lactase gene is always active, or it continues to be transcribed throughout your life if you have lactase persistence. Okay, here we are. Question 10. This looks like the last question because these comprehension questions always come up right at the end of paper 2. So they're normally worth quite a lot of marks. So I think it's really, really worth when you attempt these questions, when you do these questions, making little notes and kind of prepping yourself. Okay, so we read the passage, and we're going to read it together. Okay, so complete achromatopsia is a form of complete color blindness. It is caused by only having rods and no functional cone cells. People with complete achromatopsia have difficulty in seeing detail. Complete achromatopsia is caused by an autosomal recessive allele and is usually very rare in populations with only 1 in 40,000 being affected. However, on the Pacific island of Pingalap, 10% of the population are affected. Okay, so immediately I'm thinking, like, is this going to be Hardy-Weinberg, ? Because I've got some information about sort of like my double recessives, 1 in 40,000, or 10%, depending on the place. And then I'm also thinking about rods and cones. I'm thinking about kind of iodopsin and rhodopsin. And that's kind of where my brain is at the moment. Okay, so let's keep going. One form of red-green color blindness is caused by a sex-linked recessive allele, which affects more men than women. People with this red-green color blindness are unable to distinguish between red and green, and also between other colors. They have green-sensitive cones, but the photoreceptive pigment they contain does not function. Okay, so this is the iodopsin. Isn't working in those green-sensitive cones, and this is sex-linked. So I'm going to highlight this. Sex-linked recessive, autosomal recessive. So we're looking at the difference between autosomal recessive conditions and sex-linked recessive conditions, potentially. All right, let's keep going. Scientists investigated the use of gene therapy to correct red-green color blindness in monkeys. They injected viruses containing the genes for the green-sensitive pigment directly into the eyes of the monkeys. Although the monkeys maintained 2 years of color vision, there is debate on whether this form of gene therapy is worthwhile. I presume it ran out after 2 years. No clinical trials of this procedure have been carried out on humans. Current research into the treatment of red-green color blindness involves the use of iPSCs, induced pluripotent stem cells. The use of iPSCs could have advantages over the use of gene therapy. Use the information in the passage and your own knowledge to answer the following questions. People with complete achromatopsia have difficulty in seeing detail. Okay, well, we need to think about kind of the structure of the eye for this one. So, there's the eye. This is the lens. This is a terrible drawing of the eye. This here is called the fovea, and it has a higher density of cones. So this is where we see detail. Okay, generally we have more rods in the peripheral vision and more cones near the fovea, which is a point at which we focus light. So if the cones aren't working, if there are no cones at all, then we're going to have less resolution and we're going to see less detail. Okay, is that enough? Is that enough? Okay, so no cones and only rods. Yeah. Yeah, no, okay, nothing about the Furveyor here. This is, okay, you want to talk about how they are wired. Yeah, this is a new to me. I've only taught this part of the eye this year, so this is kind of new to me. That Furveyor stuff is true, but what they want you to say is cones are connected to a single neuron, whereas rods are connected. You normally have 3 rods connected to one neuron, whereas in cones it's one cone, one neuron, one cone, one neuron. In actual fact, it goes cone, bipolar cell, neuron, and I think rods go, yeah, three rods to one bipolar cell to one neuron. Is that right? Yeah. Okay, so we need to talk about this wiring. Moving on. 10% of the population on the Pacific island of Pingalap are affected by achromatopsia. Use the Hardy-Weinberg equation to calculate the percentage of this population who are heterozygous for the disorder. Okay, so first of all, you need to remember your Hardy-Weinberg equation. So it's P plus Q equals 1, and it's P squared plus 2PQ plus Q squared equals 1. So what have we got here? We know that 10% are affected by achromatopsia. So that's the Q squared. So Q squared is sort of 0.1. That's like equivalent to 10%. Q squared is 0.1. So I want to work out how many people are heterozygous, which is 2PQ. So if Q squared is 0.1, then Q equals the square root of 0.1. So we just do that in the calculator. 0.1, turn it sideways, square root, 0.316. And therefore P is 1 minus that, which equals, let's do plus minus plus 1 equals, so 0.8683, 684, sorry, rounding. And then 2PQ, times them together. So times 2 times 0.316 times 0.684. So times 2 equals times 0.316 equals. So that's 43%, 0.432. So 0.432. So it's 43% of the population. Okay, let's double check that my maths was correct. Yep, somewhere between 42 and 44. Okay, so that's fine. Okay, red-green color blindness affects more men than women. Okay, so this is a this is a sex-linked condition. So for men to inherit the condition, they only need to inherit 1 copy of the faulty allele. For women to inherit the condition, they need to inherit 2 copies of the faulty allele. And the reason being that men only have... So, okay, let me start again. So this is a sex-linked condition, which means that the gene is on the X chromosome. Okay, as men only have 1 X chromosome, if they inherit that 1 X chromosome with a faulty copy of the allele, they've got it. Whereas women need to inherit 2 copies of 2 X chromosomes, each with a faulty copy of the allele. Essentially, I'll draw this out. It's easier for me to draw it than to explain it. Let's do big C. No, let's big C for can see color and small c for color blindness. Okay, so for a man to be colorblind, it's just like X little c and then Y. For a woman to be colorblind, well, first of all, a woman might be like this, heterozygous, 1 normal functioning allele, 1 non-functioning allele. She would not be colorblind. For her to be colorblind, we'd have to have XC, XC. So, there's a much lower chance of her getting the double dose of the faulty allele. What do we actually need to say? Okay, it's on the X chromosome. Yeah, females require 2 alleles. Yeah, males only require 1 allele. Fine. Okay, moving on to 10.4. 10.4. Current research into the... Sorry, did I? Yeah, I missed one. People with red-green color blindness are unable to distinguish between red and green, and also between other colors. Okay, so lines 8 to 10. So let's go back. You know, it is actually, I forgot to highlight that it is worth going back and looking at the actual lines. 8, 9, 10. So we're in this region here. Unable to distinguish red and green. Also, they have green sensitive cones, but the pigment does not function. Okay, so let's keep going down. How do I explain this? So they have green. Their green sensitive cones do not detect green light. So whether they see a red object or a green object, I'm really struggling to articulate this because I think it's... So the cones, the sensitivity of the cones is like this, red, and then the green cone is kind of quite close actually, like this, and then blue is like way over here somewhere. Okay, now essentially if you see an object, the way your brain distinguishes whether it's red or green is to do with the relative amounts of the red cones and the green cones firing. So if you're getting more red cones firing than green cones firing, you see it as red. If you're getting more green cones firing than red cones, you see it as green. But what if you don't have green? Well, then there's no way of distinguishing red from green, because actually if you have an object, you know, that's here, actually we would normally perceive this as yellow. If something is bang in the middle between red and green, we perceive it as yellow, which is kind of our brain saying, okay, there's red cones firing and there's green cones firing, and so we're going to call that yellow. If you don't have green, you'll just see red as red, you'll see yellow as red, and you'll see green as red. You will only be able to kind of see red and blue kind of distinguishing. I have no idea how to put that into 3 marks, to be honest, so I'm just going to look at the mark scheme. Okay, the green cones aren't there. There are 3 different types of pigment and cone. I did explain that. So we've got red, blue, and green. So other different colors are due to the stimulation of more than one cone. Okay, yeah. Yeah, that's the mark scheme. If my explanation did not make sense, I really, really recommend checking out a video by Vsauce YouTube channel called This Is Not Yellow. It will blow your mind. I think it's great. Look it up. Vsauce, This Is Not Yellow. Really interesting 5 minutes to spend watching that video. Okay? Right. Let's move on. (10.5) Current research into the treatment of red-green color blindness involves the use of iPSCs. Suggest how iPSCs cells could correct red-green color blindness. Okay, well, iPSCs could, if they're injected into the back of the eye, the iPSCs could differentiate into rod cells and cone cells to replace defective cone cells. Now, I think they would have to be engineered. Yeah, okay, so that's right. iPS cells divide and differentiate into cone cells. Okay, now the reason I was hesitating on that is because, like, the text implies that these iPSCs would come from the patients themselves. But if a patient is red-green colorblind, and then you make stem cells from them, those stem cells are still going to have the genetic fault. They're still going to be colorblind. Like, the stem cells are still going to produce defective cones. So I think you would have to maybe take some stem cells and then genetically engineer them and then put them back into the person, is how I would think it would. You would actually have to do this. I wonder if this talks about... this here? Well, it's not that clear. This says the use of iPSCs could have advantages over the use of gene therapy to correct green color blindness. So this is talking about that part where it said the monkeys only can be cured for 2 years. So we need to think, why is that? So we could say in the trials, let's go up to the text and we'll kind of annotate. So this is this bit here. The monkeys maintained 2 years of color vision. Okay, so the gene therapy, so the virus inserted the gene for the correct green, for the correct photoreceptive pigment into the cone cells. However, those cone cells don't last forever. Okay, they are going to die and be replaced by stem cells dividing to produce new cone cells. So when those cone cells that have been, the word is transfected, that means they've received this new gene. So when those cells die, the new ones that replace them, they don't have the gene anymore. So this is why using iPSCs could be an advantage. But I think you would have to say that if you did use iPSCs, you would need to first engineer the stem cells so that they contained the new gene. So I suppose you would use the virus to infect the stem cells, that's the iPSCs, and now these stem cells are transformed stem cells with the correct gene, and then those stem cells could be put into the person's eye, and that would be a permanent cure for that person's eye, because the stem cells would keep dividing to replace themselves and to replace functioning cone cells again and again and again throughout that person's life. Okay? Right. Let's see what the marks of that is. So iPSCs long-term, single treatment, harm, side effects from use of viruses. Okay, no, so some of this, I mean, I'd probably get the long-term thing I'd got. Because they're iPS cells, there's less chance of rejection because they come from the person themselves. Less chance of immune response. I guess like repeated doses. Okay, this is another problem with gene therapy actually that uses viruses. I forgot to say. So gene therapy that uses viruses, sometimes it can be effective, like the first few times you use it, but the body then learns to, like, if you keep giving the body a virus, even though the virus is doing a good thing, the body's still like, excuse me, what is that virus? That is a foreign body in my body, and I'm going to produce antibodies against it. So the more frequently you use the gene therapy, the less effective it can become, because your body is mounting an immune response against the therapy, against the virus. So the iPSC cells is a single treatment, it's a one-off treatment, whereas the gene therapy would have to be multiple times. And I suppose there could be side effects from using the viruses. I do still think here, and it's not mentioned, that you would have to genetically engineer the iPS cells. I'm surprised I don't mention that, but that is definitely true. And I think that brings us to the end of the paper, I believe. Is that the end of the paper? Yes, it is. Okay, so I hope you find that useful. That was an interesting paper for me to do. And I think I've learned a lot about the kind of phrasing that is required, because my default mode is thinking about other specs. But I think it is really important that you kind of learn the specific phrasing that you need for each question. Practice, practice, practice. I hope you find that useful, and I'll see you next time when I put out a paper 3 run through in a week or so. Okay, thanks very much. Bye-bye.